in the amount of time that we have. So because it's a lot of chapters, if you have any questions, please type them in chat so that way like the person presenting at the time could just like go through it. And then so type them in chat. I'll be answering the questions in chat. Um, so Kelly will be going over the first half of the presentation. I'll be going over the second half of the presentation. So for the first half, type the questions directly to me in chat, and then when it's my turn to present, type the questions to Kelly in chat. And then we'll answer it as soon as we can. And then number two is that obviously we're not gonna be able to cover everything in terms of like what happens in each individual chapter. So if we don't cover something in each chapter, that doesn't mean that it's not going to be on the exam. So just use our review slides as like supplementary studying materials. Make sure to pay attention to Dr. Fat's slides as like the main studying material. And with that, I think we can start. So thanks for coming to the review session.
Hi everyone, I'm Kelly. So I'm gonna go over the first half of the review session, starting from chapter 8, energy, entropy, equilibrium, and enzymes. So there's two main forms of energy, potential energy and kinetic energy. And then in cellular energy, potential energy comes in the form of chemical bonds, concentration gradients, electrical gradients, and free energy. For kinetic energy, that comes in the form of ATP. Oh, first of all, is my slides like, is everything okay with like how you guys can see it?
Yeah,
Oh,
yeah, we can see it.
okay, perfect. Okay, so again, so kinetic energy, energy that does work, that alters the state of motion of matter, it comes in the form of ATP hydrolysis, movement of molecules down concentration or electrical gradients, and it also comes in radiant or light energy and heat. So the laws of thermodynamics applies to all matter and all energy transformations in the universe. And the first law of thermodynamics is that in converting energy forms, energy is neither created nor destroyed. So when energy is converted from one form to another, the total energy before and after the conversion is still the same. And then we have the second law of thermodynamics. When energy is transformed, some energy becomes unavailable to do work. So even though it's the same amount, the total is the same, some becomes unavailable to do work. So no physical process or chemical reaction is 100% efficient. So not all the energy released can be used to do work. So entropy, which is the unusable energy or the measure of disorder in a system, never decreases. When we have catabolism, anabolism, and metabolism. So anabolic reactions are reactions where complex molecules are made from simple molecules, and captured energy is stored in the newly formed chemical bonds as potential energy. So if you see here, we have two glucose molecules, more simpler ones, and they become one larger cellobiose molecule, so one like larger, more complex molecule. So for anabolic reactions, energy input is required, and these are energy storing reactions. Then for catabolic reactions, complex molecules are broken down into simpler ones, and energy is released from the breaking of the chemical bonds. So energy output is released. So if you see here, we have one complex one, and then the bonds are broken, and we have two more simple molecules as the product. Then we have exergonic and endergonic reactions. So exergonic reactions release free energy. This delta means delta G is the change in free energy. So in an exergonic reaction, the delta G, the change in free energy, is negative. These are catabolic reactions, and again, that means complex molecules are broken down and free energy, and small molecules are like the products. Then we have So then we have endergonic reactions also here. So this is exergonic, this is endergonic. And endergonics reactions are reactions that consume free energy, meaning the delta G change in free energy is going to be positive. These are anabolic reactions, and free energy and small molecules are used to produce a larger, more complex molecule. So one thing to note is that if A to B, so changing from reactant A to B to product B is spontaneous, then it releases energy and it's exergonic and it's catabolic. In that case, if we're talking about this reaction A to B being exergonic, then B to A must be non-spontaneous and requires or consumes energy, meaning it's endergonic and anabolic. So for equilibrium, like I just said, if a reaction is going from A to B, it can also go from B to A. So in principle, all reactions are reversible. The concentrations of A and B determine which direction will be favored. So more A speeds up the forward reaction going from A to B, and more B speeds up the reverse reaction going from B to A. At the point of chemical equilibrium, the relative concentrations of A and B are such that the forward and reverse reactions take place at the same rate. So for cellular energy, we have ATP, adenosine triphosphate. So all living cells use ATP for the capture, transfer, and storage of free energy. Let's see. ATP can be hydrolyzed to yield ADP and an inorganic phosphate. So some of the free energy released by certain exergonic reactions is captured in the formation of ATP from ADP and a phosphate group, which can then be hydrolyzed back into ADP and phosphate group and release free energy to drive endergonic reactions. So it's like a very cyclical process. Let's see. Like I said, it's a very cyclical process. The process would be ATP plus water, so H2O, a molecule of H2O. It's ATP hydrolysis. It forms ADP and a phosphate group. And in that process, it also releases energy. And then if you can see here, the change in free energy is negative 7.3 kcal per mole, meaning it's an exergonic reaction. So then with this energy, you can use ATP. Sorry, is there someone? Okay, so sorry. So then this coupled reaction has an overall negative. Sorry, the endergonic reaction after this exergonic reaction happened releases the energy. The energy is used in an endergonic reaction where the energy stored in a new PO bond is greater than the energy in an HO bond that forms after hydrolysis. So free energy is released in the hydrolysis process. And this is an endergonic metabolic reaction. So this is the formation of glucose 6-phosphate from glucose. And the formation of this is endergonic, and you can see that from the change in free energy being positive. So the first reaction releases more energy than the second reaction captures in its bond. So overall, the coupled reaction has a negative free change in free energy, meaning the overall reaction is exergonic. So for catalysts, which are enzymes which are biological catalysts. So the direction of a reaction can be predicted if the delta G is known, but the rate of the reaction cannot be predicted on the basis of just the delta G. So some reaction rates are low because there's a large energy barrier between reactants and products. And this is the energy barrier where you see like a little, like a bump in the energy required to go from reactants to products. Energy is required to surmount the barrier. This is the activation energy. Once the barrier is surmounted, the reaction goes to completion. For a stable reactant, exergonic reactions only proceed after the addition of a small amount of energy, called the activation energy here. So the activation energy increases the intrinsic or the kinetic energy of the reactant and transforms it into a transition state intermediate. Once the transition state intermediate is reached, the reaction proceeds. So this is basically what I just said, but what you should know is that more stable states are less likely to cross over the energy barrier on its own. So if you can see this versus this, a less stable state it's gonna be a lot more easily crossing the energy barrier. Okay, so there are two ways to overcome the energy barrier. So it's either one, increasing kinetic energy of the reactant, or lowering the energy barrier, which is what catalysts do. A catalyst is any substance that speeds up a chemical reaction without itself being used up or being permanently altered. So it's like a two-part definition. One, speeds up the chemical reaction. Two, it's not used up in that process. It does not change the magnitude of free energy difference between the reactants and products, and it does not affect the final equilibrium. It only changes the reaction rate. So the rate of the uncatalyzed reaction, so without enzyme right here, is directly proportional to the concentration of the reactants. The rate of the catalyzed reaction is initially high, but at some point it saturates at a maximum rate, and saturation occurs when all catalysts are bound to a substrate. So enzymes and their mechanisms. So binding a substrate, the reaction, which is the reactant in an enzyme catalyzed reaction to an active site. So the active site is a specific 3-dimensional location on the enzyme protein, and it produces, so the binding of a substrate to the active site produces an enzyme substrate complex, like ES. So specificity is when hydrogen bonding, ionic attraction, covalent bonding, or covalent bonding acting individually or together holds these complexes together. So the enzyme substrate complex generates the product P and free enzyme E. So if you see here, the equation would be E plus S. So enzyme plus substrate forms CES enzyme substrate complex, and it produces the products, so P1, P2, and the enzyme itself, which again is not used up in the reaction. So anabolic enzymes. Yeah, so anabolic enzymes help form complex molecules from multiple simpler ones. Kind of like anabolic reactions, you form one single more complex molecule from multiple simpler ones. And then free energy is consumed. So in anabolic enzymes, the active site binds two substrates to form one enzyme substrate complex, which in turn releases the product. Oh yeah, catabolic enzymes. Sorry, I forgot to mention this. So catabolic enzymes, like catabolic reactions, help break down complex molecules into simpler ones and release free energy in the process. So it's the same thing as catabolic reactions, and it's the same as anabolic enzymes, but it's that instead of this one being one more complex molecule into simpler ones, this is simple, multiple simple ones, or sorry. So breaking down one complex into simpler ones and releasing free energy. So the shape of enzymes the shape of enzyme active sites allow a specific substrate to fit kind of like a lock and key mechanism. Oftentimes orientation of the substrate and the enzyme have to line up in proper positions to overcome their natural repulsion and make them an enzyme substrate complex. Enzyme substrate complexes may have induced instability in substrates, meaning through interaction with substrates, enzymes can weaken some bonds and make them more breakable or more vulnerable to attack. There's also substrate modification. So sometimes the binding of an enzyme to a substrate can cause a chemical change in the substrate chemically by the transfer of charges or functional groups. Some of these interactions may involve transient covalent linkages between enzymes and substrates. There's also conformation changes in enzymes. So this means the change in the enzyme's 3D shape. So oftentimes the binding of a substrate can cause a change in enzyme shape or the shape of their active site, and this is called induced fit. Enzyme regulation mechanisms. So there's with enzymes, there's naturally occurring inhibitors that bind to the enzyme if there's a need to slow the reaction rates and regulate metabolism. So there's irreversible inhibition, which occurs when the inhibitor inhibits the enzyme's ability to interact with its normal substrates. This is not a super common process that happens in cells. Usually it happens when there's like poisoning, or it's like usually you don't want something irreversible. So here, this example shows acetylcholinesterase, this right here, hydrolysizing ACH into choline and acetic acid. There's also reversible inhibition, which occurs when inhibitor bonds non-covalently to enzymes and can later be released. So there's two types of reversible inhibition. There's competitive inhibition, which is when an inhibitor binds to an enzyme's active site, and it competes with their natural substrate for the binding site. Then there is non-competitive inhibition, which happens when the inhibitor changes the shape of the active site in a way that the active site no longer binds to the substrate. And then there's uncompetitive inhibition, which binds only to uncompetitive inhibitors bind only to the enzyme substrate complex, while so the difference between that is the non-competitive inhibitors can bind to both the free enzyme and the substrate, while the uncompetitive inhibitor binds changes the shape of the active site. So let me repeat. So non-competitive inhibition is when the inhibitor changes the shape of the active site so that the active site no longer binds to the substrate. And then the uncompetitive, right here, binds only to the enzyme substrate complex. We have allosteric regulation. So the change in enzyme shape and non-competitive inhibition, what we just went over, is an example of allostery, where some enzymes can assume multiple shapes. So when bound to an activator, they assume the active form, and when bound to an inhibitor, they assume the inactive form. Allosteric enzymes are controlled by allosteric regulators, and these regulators bind to an allosteric site, which is separate from the active site and change the structure and function of the enzyme. So again, that's an example of that would be non-competitive inhibition. So allosteric regulators work in two ways. There's positive regulators, which stabilize the active form, and negative regulators, which stabilize the inactive form. So for feedback inhibition, the first reaction is a commitment step. The other reactions happen then in sequence. And feedback inhibition or end product inhibition is the final product. The final product acts as a non-competitive inhibitor, so it changes the shape of the enzyme, so it can't bind to substrates of the first enzyme, so the pathway shuts down. The effect of pH on enzymes. pH can change the charges of some enzymes, carboxyl and amino groups, such as carboxyl and amino groups of the amino acids. This affects the interactions of the amino acids and can can change the structure of the protein. Temperature and pH affect the rate of the enzymatic reaction. So each enzyme is most active at a particular pH, meaning it performs the best at a particular pH. Some organisms can live at a wide temperature range. Some organisms that can live at a wide range of temperatures generate different forms of an enzyme called isozymes that function optimally at distinct temperatures. So some high temperatures can inactivate enzymes by breaking non-covalent bonds. So if the tertiary structure is disrupted by this high temperature, the enzyme is said to be denatured. So that was chapter 8. Chapter 9. So we're going to start off with so energy and metabolism. We start off with the sun providing energy for plants to photosynthesize, which then produces glucose, which is used in glycolysis, which then produces pyruvate. So once we have pyruvate, we can go into two different pathways. There's aerobic respiration and anaerobic respiration. So there's aerobic, which is with O2, and the anaerobic has O2 absent. And with aerobic, there's cellular respiration, and the anaerobic, that's fermentation. Okay, so first, before we go into that, let's go over oxidation reduction reactions. So oxidation reduction reactions is a reaction where one substance transfers one or more electrons to another substance. So it's the transfer of electrons between two substances. So electrons can be transferred as free electrons, but it's also often transferred by hydrogen ions, which is a hydrogen atom containing So a hydrogen atom contains like the hydrogen proton and a hydrogen atom. An electron atom. Hydrogen proton plus the electron. So when molecules lose hydrogen atoms, it becomes oxidized. So reduction is the gain of electrons by an atom, ion, or molecule, and this occurs to the oxidizing agent or the oxidant. So the substance that is reduced or gains electrons is the oxidizing agent, and it causes the other substance to lose electrons. So, and then oxidation is the loss of electrons. So the reducing agent is the reductant. This is the substance that is oxidized. It loses electrons, and then it causes the other substance to reduce, aka gain. So it becomes, basically, I remember it as it becomes reduced in charge. So if you gain electrons, your charge becomes more negative. So the reduction agent causes the other substance to become reduced by gaining electrons. So these two processes always occur together. So as one chemical is oxidized, the other, the electrons it loses is transferred to the other chemical, which reduces it. So one cannot happen without the other one. These are always paired. And the oxidation is usually exergonic. Reduction is usually endergonic. This glycolysis, just the meaning. So again, glycolysis, it's an enzymatic breakdown of glucose to produce pyruvate acid. It begins with glucose breakdown, and it's the first step of cellular respiration, which now we're going to go into. Okay, so the first step in glycolysis is one molecule of glucose. So ATP loses a phosphate to turn into ADP, right here, which then produces 3 molecules of G3P, which normally also creates glycerol. So then the G3P gets used, and ADP combines with a phosphate to form ATP. I'm sorry. So, the glucose becomes fructose 1,6-biphosphate, which is a 6-carbon sugar, which then produces 3 molecules of G3P. So, like I said previously. Then G3P gets used and ADP combines with a phosphate to form ATP. As NAD plus takes in a hydrogen, it becomes NADH. So up here, as you can see up here, that G3P turns into pyruvate molecules. So basically, as a summary of all these 9 or 10 steps, glucose uses up ATP to turn into fructose 1,6-biphosphate, and then fructose 1,6-biphosphate turns into G3P, and NAD plus becomes NADH, and ADP plus a phosphate group turns G3P into pyruvate. So this is the last steps for 6 and 7. So as a summary for glycolysis, 2 molecules of ATP are used up, while 4 molecules of ATP are gained during glycolysis. So 4 gained minus the 2 that are being used, there's a net gain of 2 molecules of ATP from glycolysis. There's also 2 molecules of pyruvate produced by the end of glycolysis, if you see right here, so after step 10. Okay, so that was aerobic respiration. We can now look at when oxygen is absent or there's not enough oxygen. We have fermentation. So for anaerobic respiration, it's a process that does not require oxygen O2 to convert glucose into ATP. So some cells use fermentation, which is the degradation of a substance such as glucose to smaller cells such as lactic acid or alcohol with the extraction of energy. So this is used in some cells, like I said, and pyruvate oxidizes NADH to NAD+, which converts into lactic acid or ethyl alcohol, aka ethanol. So fermentation also produces CO2, and the breakdown of glucose is incomplete, so much less energy is released when glycolysis is coupled with fermentation rather than cellular respiration. Okay, so pyruvate oxidation. So after glycolysis, pyruvate is converted to acetate, and then acetate binds to coenzyme A here to produce a molecule of acetyl CoA. So this is a compound that reacts with oxaloacetate to produce citrate, which then leads us to the beginning of citric acid cycle. So for the citric acid cycle, the starting point of this 8-step reaction is the acetyl CoA, which we just went over how it's formed. So the inputs of this cycle are acetyl CoA, water, GDP, which is guanosine diphosphate, and electron carriers, which come in the form of NAD plus and FAD. So the released energy is captured by the GDP and 2 electron carriers, the NAD plus and FAD. And the outputs of the cycle are CO2, reduced electron carriers, which are in the form of NADH, and FADH2. And then GTP, which converts ADP to ATP. So the released CO2 is what we exhale when we're breathing. That was citric acid cycle. So next, after citric acid cycle, we have electron transport chain, which is a series of coupled redox reactions. So each step oxidizes the previous step and becomes reduced. So carriers gain energy when reduced, or when they gain electrons, and release energy when they're oxidized, aka lose electrons. So for the steps of the electron transport chain, we're going to start with, so it's the movement of electrons, and starting from step 1 here up here, the NADH's electrons right here and right here. These are, again, the outputs of the citric acid cycle. So the electrons from these two molecules enter the chain through the protein complex 1. So NADH enters through complex 1, and FADH's electrons enter through complex 2. After this, the electrons from both converge into ubiquinone, which is right here. It's like shortened to Q. It's a so Q is a nonpolar lipid molecule in the phospholipid bilayer of the inner mitochondrial membrane. So after this, the Q passes the electrons to complex 3, which is the cytochrome C reductase. And then complex 3 passes to the electrons to cytochrome C here, this step. And then electrons then pass to complex 4, cytochrome C oxidase. And then the last step is the electrons are passed to oxygen, to O2, which is the final electron acceptor and the oxidizing agent. This leads to the reduction of oxygen. So the transport chain stops without oxygen, which is why our bodies need oxygen. And then the slide is just like, it's all the steps we just went over, but it's in a more like visual representation of this in the mitochondrial membrane. Okay. So now we're on photosynthesis. So photosynthesis, which is the process where light energy is from the sun, is used to convert CO2 into more complex carbon-containing compounds. So there's two major pathways of photosynthesis in chloroplasts. So there's light reactions or light-dependent reactions, which convert light energy captured by the chlorophyll into chemical energy in the form of ATP and NADPH. So this includes the light-dependent reactions includes the non-cyclic and cyclic electron transport, and it consumes H2O and water and light, and it produces ATP and NADPH and a proton, H+. So that's the first major pathway. The second one is the Calvin-Benson cycle, which is the light-independent process. It uses light indirectly, and it consumes ATP, NADPH, and H+, which is a proton, H2O and CO2, and it produces sugars. So this is a plant cell. You've all seen this before. I think the main thing you really need to know, the main difference between this and an animal cell is that there's a presence of the cell wall and the structures of the chloroplast, which includes the chlorophyll, which is where light reactions of photosynthesis occur. Again, this is where the light reaction of the photosynthesis occurs. So again, part of the light-dependent reactions include the non-cyclic, and the cyclic electron transport. So the non-cyclic electron transport is the dominant process. So it's also just a movement of electrons similar to the electron chain transport chain that we just covered. So we're looking at the movement of electrons. So step 1 is that electrons are extracted from water in photosystem 2. So over here, and note that photosystem 2, the processes occur here first, before photosystem 1. So that can be a little bit confusing, but photosystem 2 is where we're going to focus on first. So electrons are extracted from water in photosystem 2, and then it's passed to the electron acceptor molecule. And then just like in mitochondria, electron transport chain creates protons gradients, so the ATP is produced. It's then passed to photosystem 1. Ultimately, it's donated to NADP+, which is the final electron acceptor in that liquid. NADPH is the final product. And then this whole system, or this whole process right here, it consumes H2O, which is the source of the electron, and it produces NADPH, a proton, and ATP and O2. So both photosystems 2 and photosystems 1, the systems absorb photons and ejects electrons. So photosystem 2 recovers the electrons by oxidizing H2O to O2, and photosystem 1 recovers the electrons from photosystem 2. So it's just passing the electrons. Primarily, photosystem 2 absorbs light at 680 nanometers, and photosystem 1 absorbs light at 700 nanometers. Okay, so chemiosmosis is the source of ATP produced and photophosphorylation. So photophosphorylation is the electron transport coupled with transport of protons across the thylakoid membrane. So mechanisms for ATP, it's a mechanism for ATP formation in chloroplast, and it compares to chemiosmosis mechanism in mitochondria. So the steps are protons transported from stroma to thylakoid by electron transport, which creates a proton concentration gradient across the membrane, and then ATP synthase uses energy from movement of protons back to the stroma to form ATP. And then we have a knowledge check. So which sequence gives the correct order of steps taking place during the light reactions? You can just say in the chat, but if not, since we're running 4 hours on time, the answer will be B. So, sorry, D. It's D. So it goes from light energy to excited electron to electron transport, and then the pH gradient and ATP synthesis. And then the second knowledge check, electron transport and photophosphorylation provides the Calvin cycle with, so the outputs of the light dependent process, it's going to be B, ATP, and NADPH. So now we can go into the Calvin-Benson cycle, which is the second major pathway in chloroplast for photosynthesis. Okay, so cholera, which is a unicellular freshwater green algae, provided the photosynthetic system. Other products of the cycle were found by increasing the length of time and exposure in a stepwise manner until the whole pathway was revealed. Okay, so this is also called the cyclic electron transport. This is an alternative to the non-cyclic. This is needed since the next step requires more ATP than NADPH, so this produces more ATP. So the Calvin-Benson cycle, or the reductive pentose, which is 5-carbon sugar phosphate cycle, takes CO2 from the atmosphere to produce carbohydrates, and this process is also called carbon fixation or carbon dioxide fixation. So for carbon fixation, the initial reaction fixes just one CO2 into a 5-carbon compound, which is ribulose 1,5 biphosphate. This reaction is catalyzed by rubisco, the most abundant protein on Earth, and then this creates an unstable 6-carbon intermediate that quickly breaks down to form 2 3-carbon 3-PGs. The reduction of sugar reduction and sugar production. So products of the Calvin-Benson cycle are the total yield of solar energy conversion by green plants. Most of the stored energy is consumed in glycolysis and cellular respiration by the plant itself, but some of the fixed carbon also becomes amino acids, lipids, and nucleic acids. And then the regeneration of ribulose 1,5,5 phosphate. So 1 out of 6 G3P leaves the Calvin-Benson cycle to become sugar and other cellular compounds, and then 5 out of the 6 remain in the cycle to regenerate that ribulose. And then quick knowledge check, which of the following occurs during light-independent reactions of photosynthesis? Because we're running short on time, the answer is B, CO2, which is converted into sugars. Okay, chapter 11, the cell cycle and cell division. So cells divide for 3 reasons: reproduction, which is mostly in unicellular cellular organisms, growth, when organisms just their bodies get bigger, and regeneration or repair. So cell division in both prokaryotes and eukaryotes must have the below 4 events in order to occur. So the first is reproductive signal. This initiates the cell division. There's DNA replication, which is when the cell's genetic material is duplicated for 2 new cells. And then DNA segregation, which is the distribution of DNA into the 2 new cells. So each receives a copy of each chromosome. And this step, so that it ensures each daughter cell contains a complete set of chromosomes. This is a critical part of cell division. And then the last part, cytokinesis, which is when the cytoplasm divides to form 2 new cells, each with a cell membrane or cell wall. So cell wall will be implants. So we're going over prokaryotic cell divisions first. So prokaryotic cells divide by binary fission, and it results in reproduction of entire single-celled organisms that is genetically identical to the parent cell. And remember, prokaryotes have 1 chromosome. It's usually a circular strand of DNA called a plasmid. So binary fission, when this unicellular organism splits, binary fission, it results in 2 new cells. And it also occurs with the 4 steps that we mentioned earlier, cellular division cells, which for prokaryotes, it's usually external factors such as nutrient concentration and environmental conditions. So for many bacteria, abundant food supply speed up the division cycle. So as long as there's food, prokaryotes are probably going to be dividing. And then next, the DNA replication of the plasmid, there's DNA segregation, and then it also ends with cytokinesis. So the knowledge check, a bacteria cell gives rise to 2 genetically identical daughter cells by a process known as what we just covered, deep binary fission. And then bacteria usually have blank, whereas eukaryotes have blank. So as we just mentioned, bacteria, prokaryotes typically have just 1 chromosome that's circular, whereas eukaryotes have many chromosomes that are linear. So that's going to be A. Now I'm going to eukaryotic cell divisions. So we're going to start with G0 phase first, which is when the cells arrest. If a cell is not destined to divide further, it usually leaves the cell cycle. And then for interphase, which is the period between divisions where between divisions when cells grow. So in interphase, the cell grows, performs normal cell functions, it replicates DNA, and it preps for division. And then again, if it doesn't, if it's not destined to divide further, then it goes to G0. But otherwise, interphase is split into 3 subphases, G1, S, and G2. So this is the preparation for cell division. G1 is when chromosomes are single and unreplicated structures. And then between G1 going into S, between this these two phases is the restriction point, which then like the cell determines if it will continue with the division. And then in S phase, the DNA replicates and sister chromatids remain together. So S phase is very important. This is when DNA replication actually occurs in all of the like cell cycle. The S phase is the only time the DNA replicates. And then lastly, the last part of interphase is G2 phase. So the cell prepares for mitosis. Okay, and then I'll check how does a nucleus in this G2 differ from a nucleus in G1. Because it's going from G1 and then S where DNA replicates and into G2, the G2 nucleus has double the amount of DNA as a G1 nucleus, so the answer is A. So phases of mitosis. Okay, so cells resulting from mitosis are identical to parent cells in the amount and kind of DNA they have. So it's identical. The DNA is identical to the parent, and there's only one. So it produces one. So like one cell becomes two in mitosis, and they will be identical to each other. So during prophase, chromosomes become condensed, and they become more visible, and centrosomes move to opposite poles. During pro-metaphase, the nuclear envelope breaks down, the kinetochore microtubules appear and connect the kinetochores to poles, and chromosomes are gradually pushed towards the like equatorial plate, which is like in the middle of the cell. Then during metaphase, the cells are lined up in the middle. And then in anaphase, they begin to pull apart in the nuclear. And then in telophase, the nuclear envelope starts to reform. So they meet in the middle, they get pulled apart, and then they're separate, and nuclear envelope starts to reform. Knowledge check which statement about mitosis is true. As we covered, two genetically identical daughter cells are formed because you're just duplicating the exact same DNA in the same way. So same quantity and same, like, the coding is all the same. So it'll be A. Really quickly, the mitotic spindles are cytoskeletal structures composed of microtubules that move chromosomes during the segregation. So remember, in the beginning, they like attach to the chromosomes, and then during anaphase, these are the structures that pull the chromatids apart. There's polar microtubules, which extend from two spindle poles, and they overlap in the middle. And then these keep the poles apart. And then the kinetochore microtubules, which attach to kinetochores on the chromatids and to the spindle poles, sister chromatids attached to opposite halves of the spindle. So chromatid attachment and separation at the end of metaphase, cyclin CDK complex activates anaphase promoting complex APC, which activates separase, which then hydrolyzes cohesion. So separase is an enzyme that degrades cohesion only when all chromosomes are aligned during metaphase. So separase, yeah. And then we have meiosis, which is so mitosis is asexual reproduction, and so it's based on mitotic division of the nucleus and produces clones that, or the offsprings are clones that are genetically identical to the parent. So then in meiosis, which is sexual reproduction, this requires gametes created by meiosis. Two parents each contribute one unique gamete to each offspring. So meiosis is creates gametes. So these gametes are produce organisms that are not identical to parents or each other. And that is like the changing of their DNA. So your DNA is not exactly going to be the same as your siblings or your parents, and that's due to meiosis. Meiosis is also the production of one haploid or one N gamete by two N or diploid cells. It consists of two separate nuclear divisions, meiosis 1 and meiosis 2, that reduce the chromosome number to haploids. So DNA replication only occurs once, though. So even though there's two nuclear divisions, DNA replication only occurs once. And again, the products are different from the parent cell and from each other. So meiosis, like I said, occurs in two separate nuclear divisions. The first one will be meiosis 1, and second one is meiosis 2. And this is all part of, like on the slide, this is all part of meiosis 1. So prophase 1, so in prophase 1, chromatin condenses and synapses aligns homologous pairs. Chromosomes cross over in late prophase 1, right here, which is also called prometaphase. So crossing over is the exchange of genetic material between non-sister chromatids and homologous pairs. So this is what allows the offspring to be genetically different from each other and from their parent. So crossing over results in recombination or recombinant chromatids, which is the exchange of some DNA between homologous chromosomes. And now a single chromosome now contains DNA inherited from both parents, rather just all the DNA from one parent. And then in metaphase 1, homologous chromosomes pair up and align at the center, very similar to when this happens in mitosis. And then anaphase 1, homologous chromosomes, which are also now called recombinant chromosomes, separate randomly and move to opposite poles of the cell. So the act of them separating randomly generates diversity. And then in telophase 1, chromosomes gather into two nuclei, and then in some cases, the original cell divides into two. So now for meiosis 1, you started with one cell, and you end with two different cells with different genetic information from each other and one parent. Now this is meiosis 2, which also consists of prophase, metaphase, anaphase, and telophase. But for prophase 1, chromosomes condense again, but DNA does not replicate. So DNA only replicates in prophase 1, which is one of the subphases of meiosis 1. In prophase 2 and meiosis 2, DNA does not replicate again. Then in metaphase, paired chromatids align across the center of each cell. This happens 2 times because there's 2 cells. And then anaphase 2, sister chromatids separate and move to the opposite end of each cell. So independent alignment of homologous pairs in metaphase 1 and then crossing over in prophase 1 guarantee genetic info of each gamete will be different. So now that we're separating, all of these will be different. So 1, 2, 3, 4, these will all be genetically different from each other and the parent. And then finishing up meiosis 2, there's telophase 2, which is the chromosomes gathering into all the separate nuclei, and then cells divide. So in the end, the result of meiosis is that you have 4 haploid cells from one original diploid. So all of these are 4 haploid, meaning they have a half set of chromosomes coming from one original diploid cell, one cell that had the full set of chromosomes. So again, differences between meiosis 2 and mitosis. DNA does not replicate before meiosis 2. And then in meiosis 2, the sister chromatids will not be identical because of crossing over. So this is important to know. It's important to know the difference between meiosis 2, meiosis 1, and the overall difference between meiosis and mitosis. Yeah. Then I think this is the last thing. So fertilization and meiosis alternate in sexual reproduction. So both haploid and diploid bodies can undergo mitosis, but only diploid can undergo meiosis. So you think of us, most of our bodies, or like all of our bodies are made from diploid cells. So we have diploid bodies, but there are some plants and fungi that have haploid cells. So their entire bodies, or the vast majority of their bodies, are made from haploid cells. And yeah. Okay, so the last knowledge check is the major drawback of asexual reproduction as compared to sexual reproduction is that it produces less variation among offspring. So asexual reproduction, just mitosis, if you just split the cell and it's identical. So like the cells in your lungs, they're identical to each other, they're not genetically different from the next lung cell. These are somatic cells, and then for our gametes, if you're producing offspring, we want them to be genetically different. So it's better for the overall population. Okay. And meiotic errors. So there's non-disjunction, which is when homologous pairs in anaphase 1 or sister chromatids in anaphase 2 fail to separate. So remember, we have the spindles that are attached to the chromosomes. If there's any error in the attachment or the separation, this can cause non-disjunction. And then either of the errors, either in homologous pairs in anaphase 1 or sister chromatids in anaphase 2, either of these can result in aneuploidy, which is a condition of lacking. It's either lacking or excess chromosomes. So either of those options are generally harmful. So you don't want to have uh, less than or more than the number of chromosomes you're supposed to have. So in humans, trisomic, or meaning it has 3 copies for chromosome 21, results in Down syndrome. And then there's also translocation, which is when a piece of chromosome may break away and attach to other chromosomes. So trisomies, having 3 copies, and monosomies, having 1 copy, are common in human zygotes, but we have a protection mechanism. So most trisomies and monosomies for chromosomes other than 21 are lethal, and this results in many miscarriages being due to this. So miscarriage is kind of a biological mechanism to produce or to protect against non-disjunction, or like if non-disjunction occurs, you have protection mechanisms that prevent them from becoming like full humans, full adults. And then the last knowledge check. So many chromosomal abnormalities, such as trisomies and monosomies, are not observed in the human population because they are lethal and cause spontaneous abortion of the embryo early in development. So, okay. And that was chapters 8 through 11. And then Shernon will take over from chapter 12 onwards.
All right. So hello everyone again. Thanks for being here. I know this is a lot of content, but that's because your last exam actually had 1 chapter less. So this exam has a chapter extra. So once again, I know this is a lot of content, but thanks for being here. I'll be going over the second half, which is chapters 12 to 16. So, um, give me from current slide. Can you see the slide?
Yes.
Okay, perfect. Thank you. All right, so once again, I'll be talking about chapter 12 to chapter 16. So first is genetics. So in terms of genetics, you have both heterozygous alleles and homozygous alleles, but you also have genes. So genes are code for specific traits, hair color, eye color, skin color, etc. And genes are made up of 2 alleles. So these alleles can either be the same or they could be different. If they're different, they're known as heterozygous alleles. So same gene at the same locus, but they carry different alleles. You have a dominant allele and a recessive allele. And then there are also homozygous alleles. Homozygous alleles are when you carry the same alleles. So you either have two dominant alleles or two recessive alleles. So heterozygous, one of each, and then homozygous dominant is two dominant, homozygous recessive is two recessive alleles. So as mentioned here, there are certain traits that are dominant, such as a cleft chin, dimples, brown, black hair, certain traits that are recessive. So you need both alleles to be recessive in order to showcase that trait, such as gray, blue eyes, no freckles, blonde hair, etc. So genotype versus phenotype. What's the difference? So genotype is the actual genetic makeup of an individual. So when someone asks for the genotype, they're referring to the specific combination of alleles, such as uppercase R, lowercase r, etc. So genotype refers to the actual combination of the alleles, while phenotype refers to what's seen physically, the actual physical appearance of that individual. Oh, and importantly, organisms with the same phenotype can have different genotype. How? A homozygous dominant will showcase the dominant trait. Heterozygous will also showcase the dominant trait. So just because something has the same phenotype doesn't mean they'll have the same genotype, which we'll also talk about later. So here is how you do a Punnett square. So a Punnett square is how you determine the chances of the genotype for the offspring. So in this case, you have a homozygous dominant parent and a homozygous recessive parent. So it's a true breeding parent. And then when you create the Punnett square, all the children have heterozygous alleles. But when you self-pollinate the children, so you have all the children that are heterozygous, when you self-pollinate, now you have a 1 to 2 to 1 ratio or a 3 to 1 ratio. So this ratio will be important because on the test, there will be times where they'll ask for a ratio. If you have two heterozygous parents, what is the ratio of dominant to recessive kids? Or what is the percent chance you'll find heterozygous kids? So just know the ratio for have when you have two heterozygous parents. So in this case, when you have two heterozygous parents, your genotype typic ratio is one homozygous dominant, two heterozygous, and then one homozygous recessive. But your phenotypic ratio is three dominant because the heterozygous showcase the dominant allele and one recessive. So we're going to talk about Mendel's monohybrid cross. So Mendel wanted to find out if two strains of peas that have different traits, in this case, one of them is round, one of them is wrinkled, will their characteristics be blended in succeeding generations? You'll have a mixture of round or wrinkled. So when he planted a true breeding round seed and a true breeding wrinkled seed, the F1 generation was all round. However, when the F1 generation self-pollinated, you had a wrinkled seed. Why? This is similar to the monohybrid cross we just did. Uh, when you have a homozygous dominant parent, homozygous recessive parent, they create purely heterozygous children. However, when the heterozygous children self-pollinate, you have a 3 to 1 phenotypic ratio. So in this case, the hypothesis is rejected. There isn't any irreversible blending of characteristics, and recessive traits can reappear in succeeding generations. The F1 generation will always be round. However, because they're all heterozygous, there is a chance for the recessive trait to showcase in the F2 generation. So this is just showing, once again, that monohybrid cross for the F2 generation. F1 generation, you'll always have heterozygous children, but you'll always have a generation that shows the dominant trait, so all round. But just because they showcase the dominant trait doesn't mean their children will all showcase the dominant trait. As you see here, there's a 25% chance for the children to showcase the recessive trait, which is wrinkled. So the law of segregation basically states that two alleles that code for the same trait separate during gamete formation. So this homozygous dominant allele will separate from the other homozygous dominant allele. So now we're going to talk about dihybrid crosses. So dihybrid crosses are crosses that require two genes. So if it's two genes, that means it's four alleles. And so students always struggle with dihybrid crosses. So I'll tell you the way that I figure out how to create a dihybrid cross. And then also there is similar to the 1 to 2 to 1 ratio for the monohybrid cross, there's another ratio for the dihybrid cross. So as mentioned before, you were trying to find out the percent chance for children for two separate genes. So in this case, spherical or wrinkled is one of the genes, yellow or green is the second one of the genes. So how do you actually create the dihybrid cross? So the way you create the dihybrid cross, so I always consider it to be like a web method. If you can see my mouse, this S allele can bind to this Y allele or this Y allele. Similarly, this S allele can bind to this Y allele or this Y allele. So the second one, this will probably be easier to, is this easier? No. Okay, you know, I'll just stay here. So this uppercase S can bind to this uppercase Y. This uppercase S can bind to this uppercase Y. So basically, I consider this allele 1 of gene 1, allele 2 of gene 1, allele 1 of gene 2, allele 2 of gene 2. So allele 1 of gene 1 can bind to either allele 1 of gene 2 or allele 2 of gene 2. So you have uppercase S, uppercase Y, uppercase S, uppercase Y. But the same thing can happen for the second allele. This uppercase S can bind to this uppercase Y. This uppercase S can bind to this uppercase Y. Everything's uppercase here, so I mean it's the same here. So I'll talk about the heterozygous one. This uppercase S can bind to this uppercase Y. Similarly, this uppercase S can bind to this lowercase y. So your first two combinations, the first two things you'll write here is uppercase S, uppercase y, as you see here, and then uppercase S, lowercase y, as you see here. And now, so that's one of the web created. The second web is with the lowercase s. The lowercase s creates a web with this uppercase y. The lowercase s creates a web with the lowercase y. So the second two things that you write here are lowercase s, uppercase y, lowercase s, lowercase y. So essentially, you have to create two connections with each allele. As long as you're able to create two connections, then you'll be able to create your dihybrid cross. So in this case, similar to the monohybrid cross, you have a homozygous dominant for two trait parent, homozygous recessive for two trait parent. So then all the children will be heterozygous for both traits. And then when the heterozygous children self-pollinate, you have what's known as a 9 to 3 to 3 to 1 ratio. What this means, you have 9 children that are spherical and yellow. You have 3 children that are spherical and green. You have 3 children that are wrinkled and yellow, and you have 1 children that's wrinkled and green. So these are blanks because this can either be a recessive, this can either be recessive, so it's heterozygous for the overall trait, or it could show the uppercase S or uppercase Y, so it's dominant for both traits. But what this is saying, 9 to 3 to 3 to 1 ratio, for 9, you have dominant for both traits. For 3, you have dominant for the first trait, so in this case, it's spherical. The other 3, dominant for the other trait, so in this case, it's yellow. And then one of them is recessive for both traits. So when you do a dihybrid cross, you'll have 16 possible combinations. And in terms of heterozygous dihybrid cross, 9 to 3 to 3 to 1 ratio. So different alleles and variations can arise due to mutations. Once again, as mentioned before, any one individual carries 2 alleles. The multiple alleles increase the number of possible phenotypes. A wild type allele is present the most in population, so wild type is what's considered normal, and mutant alleles are present the least in population. So how do genes interact with each other? So the first one, complete dominance. In this case, white is dominant over blue. So when they have children, it'll be all white. In this case, blue is dominant over white. So when they have children, it'll be completely blue. Then there's also incomplete and co-dominance. During incomplete dominance, neither allele is neither allele is dominant relative to each other. So you can have a combination, which is blue. For codominance, they showcase both blue and white. So codominance, they're codominantly relative to each other. They showcase both allele traits. Incomplete dominance, there's a blending of the allele traits. Question. In humans, for 23 chromosomes, are there 64 alleles? So alleles refer to the genes, not the chromosomes. So chromosomes contain lots of genes, right? So chromosomes have a lot of genes, and then each of these genes code for two alleles. All right. And so now we have polymorphism. And yeah, so polymorphism, in this case, dark gray is you can have all these genotypes that have different combinations. So in polymorphism, genes can have more than two alleles. So you have uppercase C, which is dark gray. You have CCHD, which is like a chinchilla type gray. CH, which is a Himalayan gray. That's point restricted. What point restricted means is that it's temperature dependent. Based on the temperature, it can change its color, and then you have lowercase c, which is just purely albino and white. So there's a dominance hierarchy where uppercase C is greater than the chinchilla color, which is greater than the point restricted color, and then greater than obviously the recessive color. So in this case, depending on the combination of alleles, you can have a graded series of coat color phenotypes. So lastly, we're going to talk about similarly to what we said about incomplete dominance. This could be seen commonly in eggplants. So you have homozygous dominant purple fruit, homozygous recessive white fruit, but they're not necessarily completely dominant or recessive to each other. They're incompletely dominant. Because of that, they're able to combine their colors so you can have these pink fruit or pink eggplants, which are heterozygous and they combined both alleles. So now we're going to talk about chapter 13, which is DNA and its role in heredity. So first we're going to talk about Frederick Griffith's experiments. So Frederick Griffith worked with two strains of pneumonia, and he determined that a transforming principle from dead cells of one strain can produce a change in the other strain. So here you have a living S strain, the mouse died when it was injected with it. A living R strain, which is non-virulent, the mouse was healthy. So even when the virulent S strain, the one that caused the mouse to die, even if you kill it by heating, if you mix it with any living R strains, as you can see here, it's still healthy. If you mix it with any living R strains, the mouse could still die. So the dead cells of one strain, so the dead cells of the S strain, can produce a heritable change in the other strain, which would cause the mouse to die. So now we're going to talk about the Hershey-Chase experiment. So the Hershey-Chase experiment was used to determine whether DNA or protein was the genetic material. So bacteriophage proteins were labeled with 35S, DNA was labeled with 32P. And so DNA that was labeled with 32P, um, which is the protein, so they found that the radioactive element left on the phage's DNA was only in the bacterium and not in the phage, which meant that DNA had entered the bacterium. However, for the protein case, after the phage was attached to the bacterium, the radioactive element was not actually found in the bacteria. The phage's protein stayed outside of the bacteria. So these are viruses. Um, they're able to donate, or not necessarily donate. They're able to inject their DNA into living cells. So what they noticed when the DNA with 32P was labeled with 32P, they found that DNA inside the bacteria. In this case, though, when the protein coat was labeled, when the virus was mixed with bacteria, the protein coat was still found on the virus. That protein coat was not found inside the bacteria. Therefore, Hershey and Chase were able to determine that the genetic material that infects bacteria is DNA and not protein. So the structure of DNA. So in terms of DNA, you have adenosine, thymine, guanine, and cytosine. So the amount of adenosine you have is equal to amount of thymine you have. The amount of guanine you have is equal to amount of cytosine you have. And then adenosine and guanine are your purines. Thymine and cytosine are your pyrimidines. And uracil, which replaces thymine and RNA, uracil is also a pyrimidine. So in a sequence, If a sequence in one strand is 5 prime to 3 prime, AGC, TGC, TGA, what's the sequence in the complementary strand? The correct answer is D. So complementary strand will always be in the opposite direction. Why? Because DNA is anti-parallel. If you have one strand that's 5 prime to 3 prime, your second strand will be 3 prime to 5 prime. And then your second strand will be exactly the opposite of whatever's written here. So A will be replaced by T, G will be replaced by C, and etc. So that's why for this one, the correct answer is T. So on a question, if a question is on the exam, as you see here, look, C and D are exactly the same sequence. You can't just look at the sequence, you have to also note the direction. Because C, even though it's the same sequence, it's in the same direction, so it can't be the complementary strand. The complementary strand has to be in the opposite direction. So 30% of the bases extracted are adenine. What percentage of cytosine is in this DNA? So we know that adenine is equal to thymine. So if 30% is adenine, that means 30% is thymine. That's 60%. You have 40% left of guanine and cytosine. 40% divided by 2 is 20%. So the correct answer is B. Yup, 20. Good job. So now the structure of DNA. So the structure of DNA was determined through several different sources of evidence, but the main piece came from X-ray crystallography. So X-ray crystallography suggested that DNA is composed of a double helix that contains of a sugar phosphate backbone located on the outside. And then you had Watson and Crick. So Watson and Crick used model building and then combined any prior knowledge to create the structure of DNA that we know as today. So there are four main key features of DNA. It's double-stranded. For the most part, it's right-handed. It's anti-parallel to each other, which, as I mentioned before, means that one strand goes from the 5 prime to 3 prime direction, the other strand goes from the 3 prime to 5 prime direction, and outer edges of nitrogenous bases are exposed in the major and minor grooves. So, which of the following molecular models describes the structure? In this case, it is C, double-stranded and anti-parallel. So, four important functions of genetic material. What is the function of DNA? Stores genetic information, susceptible to mutations, which can cause changes in our body, is precisely replicated in cell division, and is expressed as the phenotype. So, how does DNA actually replicate? So, first, you have primase. Primase binds to your template strand and synthesizes an RNA primer. So, in this case, primase is reading the template strand from the 3 prime to the 5 prime direction, and the RNA primer is being created from the 5 prime to the 3 prime direction. So primase synthesizes this RNA primer. Once primase is once the primer RNA primer is completed, primase is released. This is just a starting point so that DNA polymerase can identify that primer and then know that it has to replicate the DNA. So once that primer is complete, primase is released, and then DNA polymerase is able to bind and then synthesize new DNA starting off of that primer. And so you have a leading strand and a lagging strand. So and you also have here, which is the replication fork. So the replication fork is the site where DNA unwinds to expose bases. So in this case, DNA helicase unwinds the double helix in order to expose the bases. And then on the leading strand is able to grow continuously. Why? Because it's growing from the 5 prime to the 3 prime direction. It doesn't have to stop every time. So DNA polymerase is able to grow continuously off the strand. And then you also have proteins that help keep the strand separated. But what about the lagging strand? The lagging strand, it has to create it from the 3 prime to 5 prime direction rather than 5 prime to 3 prime direction. Because in this case, you're going from 5 prime to 3 prime, which means you're creating the opposite. So the lagging strand is oriented so that its exposed 3 prime end gets farther from the fork, and essentially there's a cycle here that occurs. For the lagging strand, you have a primase synthesizing a primer. Then DNA polymerase fills in the gap in between these primers. So primase created a primer here, created a primer here, it's creating a primer here. Then DNA polymerase comes in and fills in these gaps. And these gaps are filled in with what are known as Okazaki fragments. So leading strand grows continuously. Lagging strand, there's a process where primase comes in, synthesizes a primer, and then it goes on synthesizes another primer, etc. DNA polymerase then comes in, fills in those gaps in between the primers with what are known as Okazaki fragments. So this is just what I mentioned before. Synthesis of the leading strand is continuous, while for the lagging strand, primers are continuously synthesized, and in between primers, Okazaki fragments are synthesized in order to help complete it. And once again, this is just showing what I mentioned. So eventually the primer has to disappear. So eventually these primers have to grow because you can only have one initial starting primer. You can't keep on having primers in the middle for your complete DNA. So what happens is that DNA polymerase 1 comes in. DNA polymerase 1 replaces each primer with DNA, and then DNA ligase comes in. So there are gaps here caused by the primer. DNA polymerase 1 replaces that primer with its own DNA. And finally, in order to close the gap, DNA ligase comes in and then glues it together. So DNA polymerase can make mistakes in replication, but cells have repair mechanisms, which are proofreading, mismatch repair, and excision repair. So for proofreading, if bases are paired incorrectly, the nucleotide is removed, and then DNA polymerase itself can add the correct ones. For mismatch repairs, so the mismatch repair mechanism detects any mismatched bases. So proteins scan the DNA, and then if there are any mismatched bases, um, it can be recognized as such, and then the base and bases surrounding it can be removed. DNA polymerase can be brought in order to add the correct one. So mismatch repair is detected by proteins. Then there are excision repairs. So excision repair works very similarly, but excision repairs are detected by enzymes. So enzymes scan the DNA for damaged bases. But specifically for excision repairs, it's when DNA is damaged by radiation, chemicals in the environment, random spontaneous chemical reactions, etc. So mismatch repair is before any modification. Excision repair is already modified existing DNA, but that DNA was damaged, such as mentioned before, by like radiation, chemicals, etc. So in this case, enzymes constantly scan your DNA for damaged bases. If it notices any, it excises it out, and DNA polymerase 1 can add the correct ones. So, how do you get proteins? So, the central dogma of gene expression: DNA is transcripted into RNA, RNA is translated into protein, and then this gene expression yields all your proteins. But there are some exceptions. Certain viruses are RNA viruses, which means they can use their RNA to encode information. They transcribe messenger RNA to transfer RNA, then that transfer RNA is translated into polypeptide. But viruses can also do that to DNA. They transcribe DNA to RNA, reverse transcribe it as well back into DNA, translate it, etc. So viruses can work in several different methods. So we're just going to talk about the natural way that proteins are created. So once again, transcription creates your pre-messenger RNA. That pre-messenger RNA gets processed to become a complete mRNA. Then that mRNA goes outside the nucleus into the cytoplasm, and then you tRNA and rRNA is also involved to help create the final polypeptide, which are your proteins. So what is RNA? RNA is similar to DNA, except RNA has an oxygen. So RNA also has one polynucleotide strand. It also contains ribose, DNA contains deoxyribose, which means it doesn't contain an oxygen. RNA contains a ribose. And then RNA also involves uracil instead of thymine. As you can see here, uracil and thymine is extremely similar, except uracil has a hydrogen group, thymine has a methyl group. And then the base pairing RNA is adenine and uracil, while for DNA it's adenine and thymine. But cytosine and guanine are the same for both RNA and DNA. And so three types of RNA are used in protein synthesis. You have messenger RNA, transfer RNA, ribosomal RNA. So messenger RNA carries the transcript of the DNA sequence to the site of protein synthesis at the ribosome. So it's similar to the coding strand of the DNA, complementary to the template strand of the DNA. And before translation happens, it undergoes post-transcriptional modification. So here, the direction of transcription is in the 5' to 3' direction. So it's reading it in the 3' to 5' direction. And as you see here, it's similar to the coding strand of DNA. So AUGC, ATGC. Once again, the only difference is T and U. So it's similar to this coding strand, but it's complementary to the template strand, as you see here. Then you have transfer RNA. Transfer RNA is extremely important as it is what contains, is what brings the amino acids during translation. So transfer RNA contains nucleic acid sequences to help create your polypeptides. There are 20 different variations of transfer RNAs, one for each amino acid. And then transfer RNA has an anticodon. So mRNA has codons, which are pairs of three bases. Transfer RNA contains an anticodon that's complementary to that pair and is able to bring whatever amino acid is necessary in. And it interacts with the messenger RNA and ribosomal RNA during protein synthesis. And then ribosomal RNA is what forms your ribosomes. So it's transcribed constantly in the nucleoli, and it forms the majority of your ribosomes in your cytoplasm. And it can also provide structural support and act as an enzyme. So later on, it helps catalyze the peptide bonds in between the amino acids during translation. So now we're going to talk about the first part, which is transcription, DNA to mRNA. So transcription materials, you have your DNA template, you have your nucleotides, and then the RNA polymerase enzyme. So within transcription, there's initiation, elongation, and termination. So initiation opens up the DNA and brings in RNA polymerase. Elongation is when nucleotides are added to lengthen the mRNA. Termination is when the mRNA, after it's completed transcribing, it's released. So how does it determine what to transcribe, how to do it, etc.? You have the promoter region. So the promoter region is a sequence of DNA nucleotides that tells the RNA polymerase where it begins, which strand is the template. So in this case, the 3' to 5' is your template strand, and which direction the RNA polymerase should move. So because the promoter is in the 3' to 5', the RNA polymerase will come in, create a RNA transcript from 5' to 3'. So initiation, RNA polymerase binds to the promoter region of a gene. So as you see here, RNA polymerase binds to the promoter region of a gene. After it binds to the promoter region of the gene, it unwinds the DNA. So it's unwinding the DNA at each initiation site, and then an elongation occurs. So RNA polymerase binds to the site of the promoter region. So here, RNA polymerase binds to the promoter region and starts to unwind the DNA, and it's moving towards the termination site. As it's moving towards the termination site, it's reading this template strand, the 3' to 5'. So it's reading the 3' to 5' template strand and then transcribing an RNA transcript from the 5' to 3' direction. So your RNA transcript is anti-parallel to the template strand. So as this polymerase is moving from the promoter to the termination site, it's creating your RNA transcript. And then finally, your RNA polymerase detects the termination site. Once it detects the termination site, it releases the transcript, it falls off, or sometimes another protein can come in and just push it off. So now you have your mRNA transcript, but it still needs to be processed and modified. So here, post-transcriptional processing and modification. So this is the structure of your eukaryotic genes. You have exons, which are expressed regions. These are your protein coding regions and sequences. Then you have your introns. Introns are non-coding regions and sequences. Introns are not expressed. So introns and exons will appear on the primary mRNA transcript. However, this is the pre-mRNA, so it'll appear here. But then the introns get spliced out because you don't want any excess when you're creating your proteins, you don't want any excess code that can mess up your amino acid sequence. So the introns get spliced out, and then after they get spliced out, your mRNA will have only the complete mRNA will have only the exons remaining, which are the protein coding regions. And not every exon codes for a different protein. Sometimes they can code for different domains of the same protein. So after the exons are spliced out, then two other things happened. So this is your coding region. An enzyme comes in, cuts the end off of the pre-mRNA, and it adds a polyadenosine tail. So a polyadenosine tail is just a tail with a bunch of adenosines. So it's added to mature mRNA in order to stabilize it. Then on the other end, a 5' GTP cap gets added to the 5' end. This allows the mRNA to bind to the ribosome outside the cytoplasm, and as the mRNA leaves the nucleus into the cytoplasm, the 5' GTP cap helps protect it from being digested by any ribonucleases roaming around. So now we're into translation. Translation is the process of RNA into protein. So as mentioned before, a mature RNA travels from the nucleus to the cytoplasm. A GTP cap binds to the 5' end of the mRNA in the nucleus, helps lead it through the pores of the nucleus, and makes sure that it isn't digested. Any unprocessed pre-mRNA remains in the nucleus. It needs to be fully processed and mature to leave. And so two key events happen during translation to ensure protein synthesis. One, your codons have to be read correctly by the tRNA anticodon, and tRNA has to bring over the correct amino acid for protein sequence. So ribosomes. Ribosomes, as we mentioned before, are made up of your ribosomal RNA. And there's two subunits, the large subunit and the small subunit. So these are extremely important. They're held together by ionic and hydrophobic forces, but when they're not used for translation, they exist separately. So within these areas, you have an E site, a P site, and an A site. So these are extremely important. These are your main sites for tRNA binding. Codon anticodon interactions occur at the P and A site. E is your exit site. So tRNA adds amino acids to the polypeptide at the P site, while the A site, your amino site, presents the next codon. So this ribosomal subunit is moving from the 5' to the 3' direction. It's moving through that transcript. So the P site polypeptide gets an amino acid gets added here, E site exits, and the A site is what reads the next three codons. So the A site eventually becomes the P site. The P site eventually becomes the E site, etc. As it moves along the mRNA transcript. So within translation as well, there's also initiation, elongation, termination. So first, initiation. The mRNA transcript has a start codon. This start codon is known as AUG. So when the start codon is found by the small subunit. So the small subunit binds to the 5' GTP cap and it moves along the transcript until it finds that AUG codon. Once it finds the AUG codon, it tells the cell that okay, now we need to start translation. So here, your large subunit is then brought forward. Along with the large subunit, the tRNA comes in with the anticodon. So AUG, your anticodon will be the opposite, UAC. And with this anticodon, your amino acid, so in this case methionine, the amino acid that correlates to the codon and anticodon gets brought in by the tRNA. So this is the P site. Remember, P is the polypeptide site. Here is where the amino acid gets added. And so these three codons are then read. So now is elongation. The A site reads the next three codons. So now A site reads the next three codons. tRNA is incoming with the proper anticodons for these three here and the proper amino acid. So the large subunit then catalyzes two reactions. So as you see here, the incoming tRNA is brought in. Then two reactions are catalyzed. First, the bond between the tRNA in the P site and the amino acid is broken apart. So right here, methionine, the peptide bond between methionine and the tRNA is broken so that another peptide bond can be formed between methionine and proline. So once again, the bond between methionine and the tRNA at the P site. So I'm saying methionine, but any amino acid here, the bond between the amino acid and the tRNA at the P site gets broken apart so that eventually when this tRNA at the A site moves into the P site, a peptide bond can be catalyzed between both amino acids, and this will continue on. The proline will then break apart from this tRNA, and then whatever tRNA comes in at this A site, the proline will then bind to that amino acid at that A site. And so this ribosome is just moving along, extending that polypeptide chain with amino acids. Finally, there's termination. Uh, the ribosome reads a stop codon. When that ribosome reads the stop codon, a release factor then binds to the complex. So in this case, stop codon is UAA. The release factor comes in at the A site, binds to the complex. Then that release factor disconnects the polypeptide from the tRNA at the P site, and then that tRNA releases from this polypeptide that was created. So now your amino acid polypeptide chain is formed, and then the remaining components also separate. So after the polypeptide chain is formed, it's not just left alone. There's also still post-translational modifications that can happen. So first is proteolysis. Proteolysis is the cutting of a long polypeptide chain into its final products through enzymes known as proteases. So one gene can produce multiple peptides. So as you see here, this super long polypeptide chain is proteolysed into three different peptides. There can also be glycosylation. Glycosylation is the addition of sugars to form glycoproteins. And there's also phosphorylation. Phosphorylation is the addition of any phosphate groups catalyzed by protein kinases. And the phosphate groups can also change the protein conformation, make it secondary, tertiary, etc. So obviously our body is still prone to mutations. Mutations can still happen in our body. This is wild type. This is just normal what usually happens. You have also what are known as silent mutations. So as I mentioned before, each amino acid correlates to three specific bases. However, some amino acids can correlate to more than one combination of bases as a whole. So here in this case, proline is translated by CCC. So the combination of three cytosines gets you proline. But in this case, the combination of CCU also gets you proline. So there's no change in your amino acid sequence because there's obviously, so certain amino acids, as mentioned before, there can be multiple combinations of bases that can lead to the same amino acid. So in this case, it's a silent mutation. Whatever mutation happened, you still have the same amino acid sequence. Then you have a missense mutation. A missense mutation is where uh, a mutation happens here. So in this case, uh, you have A instead of T. That T then gets turned into U. So in this case, now you have an amino acid change at position 5. Before it was asparagine, now it's valine. So because of this amino acid change, it creates a completely different amino acid sequence. So once again, asparagine turns into valine. Then you have a nonsense mutation. A nonsense mutation is a mutation where you create a stop codon. So in this case, UGG typically gets you tryptophan. Here, UAG creates a stop codon. So now you have no protein made. The polypeptide that was supposed to be made, you only have one amino acid. So nonsense mutation creates a stop codon, stops any formation of a protein. Lastly, there's a frame shift mutation. You should have a stop codon here. Oh, However, due to an insertion of another amino acid in between bases, now everything gets shifted from that position. And so all your amino acids are changed from the point of that insertion. So here, your first two are the same. And what should have been a stop codon here, because of the frame shift mutation, now you're just creating a super long polypeptide chain that first off, the amino acids have changed. And second off, where there should have been a stop codon, now that stop codon is not there. And so this is just showing what can happen. So A is obviously just your normal functional protein. B, you still create a functional protein because it is a silent mutation. C, there can be loss of function mutations. And D, gain of function mutations. For loss of function mutations, whatever type of mutation it was, whatever protein that came out of it, it lost its function due to that mutation, whether it was a frame shift, nonsense, missense, etc. Or the missense mutation or frameshift mutation can create a new function, which is a gain of function mutation. It can code for a protein that gains completely new functions than what it was originally. So now we're on to the last chapter, chapter 16, regulation of gene expression. So, in what ways can a cell shut off supply of a protein? It can downregulate mRNA transcription. Downregulation is essentially the process by which a cell decreases the quantity of any cellular component. So, it decreases the quantity of whatever protein is going to be transcribed. It hydrolyzes mRNA, preventing translation. It can prevent any mRNA translation at the ribosome. After the protein is made, it can also hydrolyze the protein, or it can inhibit the protein function. So, in terms of what the body prefers to do, the body prefers to block transcription because it's more efficient than transcribing the gene, translating the message, and then inhibiting the protein. So, the body prefers to block transcription in the first place. So, types of gene regulation. Where does gene regulation begin? So, you have what are known as negative regulators. Negative regulators bind to the repressor binding site and it prevents transcription. So, here there's a binding site. Typically, transcription occurs. The regulator binds to it, no transcription occurs. Or you could have positive regulators, which in this case it's an activator. The binding site, typically there's no transcription. However, the regulator binds to that site. So now it's a positive regulator. It allows for transcription to occur. And so there are two ways to regulate a metabolic pathway: allosteric regulation and transcriptional regulation. In terms of allosteric regulation, it's super quick. So it allows very rapid fine tuning because only the end product feeds back. So whatever end product is created, it feeds back and inhibits the activity of only enzyme 1. So allosteric regulation can quickly block the pathway. However, transcriptional regulation, it blocks the regulation of enzyme concentration at each individual point. So transcription regulation blocks the transcription of each individual gene, which then creates each individual enzyme. And then because it blocks the transcription of all 5 genes, no enzymes are produced, no end product. So although transcriptional regulation is slower, it helps make sure that it's more fine-tuned for each specific enzyme. So allosteric regulation is very quick. Transcriptional regulation is slower, but it looks at each individual gene, each individual enzyme. So now we're going to talk about E. coli, glucose versus lactose. So typically E. coli cells like metabolizing glucose. But what happens if there is a low concentration of glucose or absence of glucose? In that case, E. coli would switch its metabolism to metabolize lactose. So obviously the cell has to eat something or else it'll die. So since E. coli doesn't like metabolizing lactose, it prefers to metabolize glucose, it needs extra help. So it needs extra help from these 3 enzymes: beta galactoside permease, beta galactosidase, and beta galactoside transacetylase. So permease is a carrier protein that helps move lactose into the cell when it normally does. It's normally inactive. Beta galactosidase hydrolyzes lactose, and then transacetylase transfers acetyl groups from acetyl-CoA to galactosidates to help make it active. So in this case, lactose induces the synthesis of beta galactosidase. So there is a lag period, but eventually after that lag period, when lactose is added to the growth medium, beta galactosidase, as you see here, the level increases significantly in order to help ensure that lactose can be brought in and hydrolyzed. And so as you see here, effective inducer on mRNA level. When the inducer is added, so in this case, the inducer is lactose. When the inducer is added, the level of lactose mRNA, in order to break it down, is then increased significantly, as you see here. But once you remove the inducer, once you remove lactose, then it returns back to normal, which is the basal level. So you have a lac operon of E. coli. So a typical operon consists of a promoter, two or more structural genes, an operator, which is a short stretch of DNA between the promoter and the structural genes. And then some DNA sequences, like operators and promoters, don't encode proteins. They're just binding sites for other proteins to help establish it later on. So in this case, here, typically when lactose is absent, there's an active repressor on the lac operon of E. coli to help make sure that obviously beta galactosidase, everything else, beta galactosidase or the other enzymes, beta galactoside permease, etc., aren't actually being formed. So there's an active repressor on it. But in this case, oh, sorry, give me 1 second. Okay, so in this case, the repressor protein has two binding sites, one for the operator, one for the inducer. So now lactose is present. When lactose is present, the inactive repressor binds to the inducer, which is allolactase. That causes RNA polymerase to then bind to the lac operon. So as as seen here, the repressor is usually active and then inhibiting any RNA polymerase transcripts being formed. But allolactase binds to the inactive repressor, which causes the repressor to detach from the transcript. As it detaches from the transcript, now RNA polymerase can create whatever RNA polymerase can bind to the transcript, create its own mRNA transcript, and then translate it in order to create your enzymes necessary to metabolize lactose. So now there are a difference between two types of operons. You have inducible and repressible symptoms. So for inducer symptoms, inducers are often the substrate of a metabolic pathway. They interact with regulatory uh, proteins, which are repressors, and repressors can no longer bind to it, allowing transcription. So in repressible symptoms, which are co-repressors, they bind to regulatory proteins, which then bind to the operator and block transcription. So inducible systems allow transcription, repressible systems block transcription. So once again, inducible systems, they help prevent the repressor from binding. Repressible symptoms, they help make sure the repressor actually binds to the operator to block transcription. So transcription in prokaryotes versus eukaryotes. So in prokaryotes, the functionally related genes are often clustered, while in eukaryotes, they're distant from each other, but they're still able to bind due to the DNA, what's it called? Sorry, folding in on itself. Then RNA polymerase, uh, there's one RNA polymerase in prokaryotes, while eukaryotes have three RNA polymerases. One is responsible for transcribing rRNA, two is responsible for transcribing messenger RNA, three is responsible for transcribing transfer RNA and small RNA. Give me 1 second. I have to just give me one. Okay, sorry about that. Prokaryotes also have very few promoters as they're less diverse. Um, Eukaryotes have many promoters, many regulatory sequences, and in order to initiate transcription for prokaryotes, RNA polymerase has to bind to the promoter. Eukaryotes bind to many proteins, including RNA polymerase. So initiation of transcription in eukaryotes. So here you have what's known as TF2D. So which is the first general transcription factor. It binds to the promoter at the Tata box. So what is the Tata box? The Tata box is a DNA sequence that indicates where a genetic sequence can be read and then eventually decoded. So TF2D binds to the Tata box, and then once it does, another general transcription factor binds to it. Then once that general transcription factor binds to it, more and more transcription factors bind to TF2D and that Tata box region. So finally, once all of these are added, RNA polymerase 2 binds after everything has been added, and now it's finally ready to transcribe RNA. So even among that promoter region, you still need TF2D and then more transcription factors to bind to the Tau box of the promoter region to allow RNA polymerase 2 to come in and then transcribe your RNA. So, as I mentioned before about DNA folding in on itself, so as you see here, you have sequences that can bind for specific transcription factors, but they could be far from the actual transcription start site. Like you see here, this is the promoter region, this is an enhancer, but the sequence is very far from each other. However, what allows for that to actually happen, what allows for the DNA to actually make sure that the enhancer region is still transcribed and make sure that transcription factor is still allowed for RNA polymerase to come in is DNA bending. So DNA bending allows transcription factors to help interact with the RNA polymerase complex and affect the rate of transcription. So as you see here, it folds in over of itself to allow for the enhancer region to bind to the complex so that RNA polymerase 2 can continue forward. So how do viruses regulate their gene expression? So you have virions, which are acellular. They're usually composed of only nucleic acids and a few proteins. And then you have host cell viruses that help carry out metabolic functions while they reproduce. So the first is the lytic cycle. So the lytic cycle is a strategy for viral reproduction. A promoter in the viral genome binds to a host RNA polymerase, and adjacent viral genomes are transcribed. So here, virus releases its DNA into the cell. So the early gene, promoter binds to the RNA polymerase. Any adjacent viral genes are then transcribed. But the late genes, these help encode the viral capsid proteins and enzymes to then eventually lyse the host cell, which is burst the host cell, and then release more of these virions through the body. So as you see here, there's also the lysogenic cycle. So these two work in conjunction with each other. So they both have the same start point. Bacteriophage binds to the bacterium. Phage DNA enters the host cell. As mentioned in the lytic cycle, host DNA is digested. New phage DNA forms from transcribed from the early genes, and then the late genes help produce these phage proteins and viral capsids. Eventually, as the assembly of the phage is completed, the enzyme causes the cell to lyse, and then the cycle starts again. But the lysogenic cycle can also occur if the host cell is strong enough. If the host cell is strong enough, the phage DNA is able to integrate itself within the actual host DNA. So here, the host DNA is digested, but in the lysogenic cycle, the phage DNA is actually able to integrate itself within that host DNA. So it integrates itself and it becomes a non-infective prophage. It then replicates eventually, and it stays within that cycle continuously until there comes a time where the prophage will excise itself from the host chromosome, and then the cell would eventually enter the lytic cycle and then burst to release more virions. So what controls if something is in the lytic or lysogenic cycle? So in a rapidly growing host, CRO synthesis is low, and then your CL protein is able to accumulate. If your CL protein is able to accumulate, you're entering the lysogenic cycle. As you see here, CL protein represses the lytic cycle, activates the lysogenic cycle, allows the viral DNA to replicate itself within the host cell over and over without bursting it. But under extremely stressful conditions, CL protein uh, is degraded, and then if CI oh sorry CI if CI protein is degraded, then CRO protein will then become more activated. More CRO protein is produced under extremely stressful conditions. As more CRO protein is produced, it then activates the lytic cycle, activates genes for lysis, causing, as you see here, in rare cases which are under the stressful conditions, CRO protein causes the prophage to excise itself from the host chromosome and enter the lytic cycle. So what about regulation after transcription? You have control mechanisms such as alternative splicing of pre-mRNA, microRNAs, translation repressors, and regulation of protein breakdown. So alternative splicing is a mechanism for generating proteins with different functions from a single gene. So as we mentioned before, when you have your pre-mRNA, you excise the introns and then keep only the exons. However, you can create proteins with different functions during the splicing. So your mRNA can have a different combination of the exons. So in this case, protein 1, it's missing exon 3, one only has 1, 2, 4, 5, 6. Protein 2 only has 1, 3, 5, 6. Protein 3 has 1, 3, 4, 5, 6. So you can mix and match the different exons that create the mature mRNA transcript by mixing and matching the different exons from the same transcript or from that single gene. You can create proteins that have completely different functions due to having different exons being transcribed. Then you also have microRNAs, and then small interfering RNAs. So proteins guide microRNAs to target your messenger RNA. In this case, when the microRNA is created and then guided to that messenger RNA is then eventually degraded, translation is inhibited. For small interfering RNAs, they result in the silencing of RNA. They typically come from viral infections and transposon sequences, and they degrade specific mRNAs. For microRNAs, they inhibit the translation of several mRNAs. So it's not just a specific one, it's several of them. While for small interfering RNAs, it degrades very specific messenger RNAs. So translation repressors, there are multiple ways to regulate mRNA translation. As we mentioned before, microRNAs can inhibit translations. You also have silencing RNAs, but then you also have repressor proteins themselves. Repressor proteins can block translation directly. So when iron is low, a translator repressor binds to ferritin mRNA, which helps make sure ferritin isn't created to help make sure that you have a higher iron concentration in your body because ferritin takes up iron. However, when it is present at high concentration, iron is able to bind to this repressor to allow ferritin to continue to be made. And then lastly, as we mentioned about regulation of protein breakdown, you have proteasomes, which break down proteins. You have ubiquitin, which attaches to proteins targeted for breakdown. So as ubiquitin is attached to the protein, that target protein is then recognized by a proteasome. Ubiquitin is then released. That proteasome is able to hydrolyze that target protein and then break it down. And that's everything. I know that was a lot of materials. This everything that we went over today should be posted on the YouTube channel by the end of day, most likely. Please fill out the feedback form. Your feedback is very valuable. Once again, sorry for the long time, but it is a lot of material as exam one had less material. Hopefully, it was helpful for you guys going over everything. Yeah, so please fill out the feedback form. If you haven't filled out the attendance form, please fill out the attendance form. Thank you so much.